It will be open from 4 p.m. and probably close at 11 p.m. And so it will cover the material that was evaluated on quiz number 2, as well as what we covered thereafter, which include the central dogma of molecular biology. We're talking replication, transcription, and translation. So those will be covered in the test on Thursday. So what we will be discussing today will not be on the test. Is that clear? All right. Any questions before we start? Go ahead, Lourdes.
Good morning, sir. I'm just wondering, when would you call us in to do the quiz? Because I really would like to do the quiz before the test. That would be Thursday.
I will see if Wednesday morning is a possibility. All right?
Okay, sure, no problem.
I will try my best to make it Wednesday morning. And this goes for others who have that evaluation pending as well. Okay, so I'll try my best for it to be Wednesday morning. If not, we'll see. If probably Wednesday afternoon, we'll see.
Okay, thank you.
Okay. Ashley says Wednesday morning she has a class. All right. Are you free Wednesday morning, Lourdes?
Well, sir, I have a class as well, but I was going to miss that class for your assessment.
And what about Wednesday afternoon?
My last class ends at 1:45, so I'm free from 2 onwards.
Okay. We'll see how it goes. All right. Control of gene expression. When you hear this term gene expression, what comes to mind? Anybody? You mentioned it last class.
Phenotype?
Say that again, Madison.
The phenotype of an organism.
The phenotype? Yes, the phenotype is very much involved in gene expression. What else comes to mind before even reaching the phenotype? Recall I said that you can have a gene present, but it is not expressed. So a gene could be present and it is not expressed for various reasons. Could be in a dormant stage. Probably it's not expressed because of the developmental stage of the organism. All right, so take for example, human beings. A male in all its cells would have the gene that is responsible for growing beard and mustache. But does a 5-year-old has beard and mustache? So you understand? But the gene is there, just that it's not expressed because of the developmental stage of that particular organism. Is that understood? And this is true for plants as well. All right. So all genes are there, but some genes are not expressed depending on the developmental stage of that particular organism. All right. So when we talk about gene expression, a lot has to do with whether transcription is taking place at that particular time. So even though the gene is there, but no transcription is taking place, so no messenger RNA is being produced, then there is no expression. So you can actually measure gene expression based on the presence or absence of messenger RNA, and subsequently based on the presence or absence of the protein it will be producing. Is that clear? All right. In multicellular eukaryotes, it is critical for directing development and maintaining homeostasis to the conversion of genotype to phenotype. So the flow of information from DNA to produce functional proteins that control cellular activities. That is the central dogma of molecular biology, which we dealt with over the past few classes. All right. In prokaryotes, the primary function of gene control is to adjust the cell's activities to its immediate environment. Changes in gene expression alter which enzymes are present in response to the quantity and type of available nutrients and the amount of oxygen. So almost all of these changes are fully reversible, allowing the cell to adjust its enzyme level up or down in response to environmental changes. Now, this is a fundamental difference between prokaryotes and eukaryotes, because most eukaryotes, they try and maintain that homeostasis, whereby the internal environment is kept constant. All right. While in the case of prokaryotes, their metabolic activities and so on adjust according to the environment that they're in. So there's a difference there between prokaryotes and eukaryotes. The cells of multicellular organisms, in contrast, have been shaped by evolution to be protected from transient changes in their immediate environment. So most of them experience fairly constant conditions. So indeed, homeostasis, which is the maintenance of a constant internal environment, is considered by many to be the hallmark of multicellular organisms. Cells in such organisms respond to signals in their immediate environment, such as growth factors and hormones, by altering gene expression, and in doing so, they participate in regulating the body as a whole. Some of these changes in gene expression compensate for changes in the physiological condition of the body. Others mediate the decisions by actually produce the body, ensuring that the correct genes are expressed in the right cells at the right time during development. All right, so earlier we talked about that. It all depends on the developmental stage of the organism, okay? So that's gene expression. Let's start a new topic, which is biotechnology. Probably heard the term before already. We will learn a little bit more about biotechnology over the next day or two. So this is the process of combining genetic material from two different sources into a single DNA molecule. And this molecule would be referred to as a recombinant DNA molecule. I have mentioned this in class before, and I've used a couple examples of recombinant DNA molecule. Anybody recall the examples I used in class?
Wouldn't the example be the insulin, the production of insulin?
Exactly, insulin is one. Whereby the gene, you see where it says genetic material from two different sources. So in the case of the insulin, you had the gene which comes from a human cell, that gene that is responsible to produce insulin, and that is spliced into a plasmid from the E. coli, a bacterial cell. And so that plasmid that now has the human gene in it is a recombinant DNA molecule, because it is combining genetic material from two different organisms, human being as well as the bacterial cell, okay? So that is recombinant DNA technology, combining genetic material from two different organisms. Yes, it involves taking a piece of DNA from an organism and splicing it into a clonal vector. Remember what this term means? Anybody recall what splice means? To splice?
Together, sir, to combine them.
To join, right? To bring together. All right. So when you talk about splicing two segments of a DNA molecule, is actually joining them together. All right. So splicing it into a clonal vector, which is a DNA molecule capable of replication in a host organism. So basically, in the case of insulin, the E. coli is actually used as the cloning vector. All right. So the E. coli itself is actually used as a cloning vector because when you actually, so you have the chromosomal DNA, and then you have the plasmid, and then the plasmid has that gene, the insulin gene. All right. So when this bacterial cell replicates, it is also replicating this foreign DNA. All right. So the bacterial cell is actually the cloning vector in this particular case. All right. So this is the cloning vector, which every time it replicates that foreign DNA. And of course, if that gene is being expressed, it will be producing the insulin. So reproduction in the host cell results in the replication of the recombinant DNA molecule. And this is what is referred to as molecular cloning. All right, so molecular cloning has to do with building a recombinant DNA molecule and put it in a cloning vector and have that cloning vector make more copies of your recombinant DNA molecule, okay? So here we have the case that Lourdes mentioned in the case of insulin, all right? So the human gene cloning, this is a human cell that we see here, whereby the gene, this right here, that gene that is responsible to produce insulin, that is actually cleaved out. The word cleave means to cut, all right? It is cleaved with restriction enzymes. So restriction enzymes that act as molecular scissors cleave that insulin gene from the human cell. And then you have this bacterium here on the right, whereby the plasmid is extracted, and the plasmid is also cleaved with restriction enzymes, all right? And then this insulin gene is inserted right in there, and it is sealed by DNA ligase. And so you end up with a recombinant DNA molecule because it contains genetic material from the bacterial cell as well as from the humans, right? So this is recombinant DNA technology. So I mentioned restriction enzymes, which are also known as restriction endonucleases, all right? So restriction enzymes or restriction endonucleases. These are required to produce recombinant DNA molecules because remember that these, whenever you hear the term restriction enzymes, think about them as molecular scissors capable of cutting nucleic acids at specific locations, okay? It says these are capable of cleaving. Remember this term, right? Cleaving means cutting. All right, so these restriction enzymes are capable of cleaving double-stranded DNA molecules at specific nucleotide pair sequences called restriction sites or restriction enzyme recognition sequence. So different restriction enzymes can recognize different restriction sites and cleave that DNA molecule right there, okay? They are named from the organisms from which they are isolated. So all these restriction enzymes are actually isolated from different organisms. Generally, a 3-letter system is used, and it is usually italicized followed by a Roman numeral. So we have some examples of restriction enzymes. So when you're reading literature, you'll be able to identify a restriction enzyme because it will normally have 3, 4 letters that are italicized followed by a Roman numeral. So I have a BGL2 here. This restriction enzyme was isolated from this particular organism, Bacillus globigi, right? EcoR1, which is a very common restriction enzyme, EcoR1, this was isolated from E. coli. And then you have HindD3, all right? These were isolated from Haemophilus influenza. So all of these restriction enzymes are capable of recognizing different locations on the DNA molecule. All right, I'll give you some examples in just a few, where on that DNA molecule they are able to recognize and cleave. There are two classes of restriction enzymes. You have type 1 restriction enzymes. These one recognize a specific nucleotide pair sequence in DNA and then cleave the DNA at a non-specific site away from that sequence. All right. So it recognizes a specific nucleotide pair sequence in DNA, but then goes and cleave somewhere else. And because of this, type 1 is not very useful in the construction or analysis of recombinant DNA molecules. You can't have a restriction enzyme recognizing a specific site, but then go and cleave somewhere else at random. When you are constructing a recombinant DNA molecule, you have specific locations that you want to be cleaved, because there is where you're going to insert your foreign gene to create that recombinant DNA molecule. Type 2 restriction enzymes. They also recognize a specific nucleotide pair sequence in DNA, but in this case, they cleave the DNA within that same sequence where they recognize. So in the case of type 2, that same location that they recognize, there is where they cleave. All right? And so these are very valuable for the construction of recombinant DNA molecules. So type 2 restriction enzymes are very valuable in the construction of recombinant DNA molecules. Some enzymes, all right, such as these ones right here, these are some examples of restriction enzymes. They would cut both strands of DNA between the same two nucleotide pairs to produce what is called blunt ends. So blunt ends are when the restriction enzyme cuts the DNA molecule straight down, one straight cut down on both strands. All right, so if you have a strand like this, these particular restriction enzymes would cut this molecule straight down like this, creating what is called blunt ends. So after the cut, you would end up with a molecule like this, right? Because the cut was straight down on both strands of that DNA molecule. All right? So these type of restriction enzymes produce what is called blunt ends. However, you have other restriction enzymes such as these ones. These make staggered cuts, and these produce sticky or staggered ends. These can have the overhanging 5' end or the overhanging 3' end. What this means is that they would cut one strand at one location, but don't go straight down. They would go and cut the second strand at a different location. All right, so it creates what is called sticky ends or staggered ends. All right, so you could probably end up with something like this. All right, so these would be sticky ends or staggered ends because it cuts the top molecule here, but not the top molecule, the top strand. And then the second strand at the bottom, it would cut it at a different location, creating what is called sticky ends or staggered ends, okay? So here, this diagram is depicting the two types. So the first two, these first two, produce what is called blunt ends, right? So blunt ends. Take note that they have the capability of recognizing anywhere there is a G and a C. So if there is a G and C on that DNA molecule, these two restriction enzymes will recognize where that is and cut right between the G and the C. And of course, G pairs with C and C pairs with G. So the complementary strand will be the CG. And hence the reason why it would cut straight down, creating what is called blunt ends. All right. And now when you take a look at the other restriction enzymes, like these other three at the bottom here, BAMH1 recognize where there is GG. So wherever there are two G's consecutive, there is where BAMH1 will go and cleave. So if you notice, it cleaves the GG here on the 5' on the top strand, right there. But then it encounters a GG on the complementary strand in a different location, right here at the bottom, all right? So it creates what is called sticky ends or staggered ends. Likewise, you have HinD3 that recognize wherever there are consecutive A on that DNA molecule, and there is where it cleaves. So it would cleave the top strand right here between these two A, and then find the two A's on the complementary strand and cleave there. Okay. EcoR1 would normally cut between A and G. So wherever on that DNA molecule there is an A and a G together, there is where EcoR1 will recognize and cleave, because remember these are type 2 restriction enzymes. So the same site that they recognize, there is where they are going to cleave. So here you have the GA at this top strand, but the AG at the bottom strand in a different location, creating sticky ends or staggered ends. All right, so you have restriction enzyme at the top, the double-stranded DNA. All right, this double-stranded DNA, and it is cleaved by the restriction enzyme. So if you notice, the top strand is cleaved at one location, and then the bottom strand is cleaved at a different location, right, between the G and the A. Okay, so this is what is referred to as sticky ends or staggered ends. Questions? All right. Let's move on to cloning vectors and cloning. So I mentioned earlier that cloning vectors are very important in terms of being able to clone certain segment of DNA. All right. So we will look at different cloning vectors and how you go about cloning DNA. So cloning refers to generating many copies of a DNA sequence. Okay. So you can generate many copies of a particular DNA sequence. To clone a piece of DNA sequence, two things are needed. A disabled host that has no significant chance of surviving outside the lab. You can't use a particular host that might have some detrimental effects to the environment, whereby if that particular organism escaped from the lab, it would create a disaster, all right? Could create maybe the next pandemic. So you want a disabled host that has no significant chance of surviving outside the lab. You also need that cloning vector, all right? A cloning vector is a DNA molecule capable of replication in a host organism and into which a gene or a piece of DNA can be specifically inserted at known positions. All right, so a lot of times we will look at the different types of cloning vectors, all right? And we will see how you actually go about building these cloning vectors, because these cloning vectors are built with the use of restriction enzyme, as well as with the foreign DNA molecule that you would like to clone. All right, so different vectors are used for this particular purpose. What types of vectors used for cloning DNA sequences in bacterial cells? So these are in bacterial cells. Plasmid cloning vector. And this one, we had the example of the E. coli being transformed with the gene for insulin. So in that particular case, remember that what was done is that the plasmid was removed, was cleaved, and with DNA ligase, the gene for insulin was inserted into it. So that plasmid now is used as that particular cloning vector to produce more of that insulin gene that you want to clone. So remember that whenever these bacterial cells are replicating, they don't only replicate the chromosomal DNA, but all plasmids also replicate in an equal proportion. All right, so plasmid cloning vectors. A plasmid is a DNA molecule that is separate from and can replicate independently of the chromosomal DNA. All right, they are double-stranded and in many cases circular. Plasmids usually occur naturally in bacteria. All right. There was another case of that I mentioned in class whereby plasmid was used to create a recombinant DNA molecule. Anybody recall that example? I can give
Isn't it the example? Sorry, I was saying for transformation of the bacteria.
Transformation of which bacteria?
The E. coli, I think.
The E. coli is actually used for insulin. All right. So when you actually insert that in the insulin gene into that plasmid, you are genetically modifying that E. coli. But in class, I had mentioned another example using plasmid as the cloning vector to transfer a foreign DNA to a different organism. Anybody recall that example I used in class? I can give you the name of the plasmid. The plasmid was called PI plasmid. What was that example? The one with the tumor. What about the one with tumor, Johara? Actually, TI stands for tumor inducing. So yes, it induces a tumor. What was that example? Transferring gene from where to where? Inter-kingdom conjugation. You are giving me some vague responses. I want a little bit more. Johara is right. The one with tumor, TI stands for tumor inducing. Kaling is right. Inter-kingdom conjugation between organisms from two different kingdoms. I need more. What was the example? With a plant, yes. Still vague. Maybe when everybody put their little 2 cents together in the chat, then we can probably tie everything together and come up with a comprehensive and complete story. One says tumor, keyword. Next one, inter-kingdom conjugation. Next one has to do with a plant. Can anybody describe the example I used that you all of you are referring to there? Can you give me in one sentence, or probably one or two sentences, the example I described in class, which is an example of plasmid cloning vector? I think last class somebody had asked me to explain that, and I did.
Sorry, sir. Isn't it the Acrobacterium 25 something facies? That's when the bacteria enters the plant cell and it induces a tumor. Oh yeah, I have it in my notes. The tumor-inducing plasmid, it enters the plant and then it induces a tumor, and I think it kills the plant cell. Something of that sort.
The bacteria enters the plant cell?
Plasmid.
Oh, and it's not the entire plasmid that actually enter the plant cell. What actually enter the plant cell? There's a portion on the TI plasmid that actually enters the plant cell. In that it says the tDNA. So the tDNA enters the plant cell. Okay. So here we have an example of a bacterial cell, and we have the chromosomal DNA, which is on the left, this portion right here. And these small circular molecules, these are different plasmids found in this bacterial cell. All right, so these plasmids are the ones that you can actually be used as cloning vectors. So you can actually take out one of these plasmids and with the help of restriction enzyme, cleave them at certain points, and then you insert foreign DNA into them, and then you have these plasmids acting as your cloning vectors. So every time this bacterial cell replicates, it is also replicating that recombinant DNA molecule that you created. Okay? So look at this. This is a plasmid, right? What you see here is a plasmid as well. There are several things that you can observe on this plasmid. What do you think all these things are that you see here on the side? These are what? What do you think these are? And it says it indicates where on the plasmid these are located. All right. So for example, here you have one in this yellow region. Here you have another one, and so on. So these portions here that I'm marking on the plasmid, what do you think these are? So it has a series of restriction enzymes that are outside. All right, so all of these are restriction enzymes, and it is indicating on the plasmid where these restriction enzymes would recognize, and so these would be recognition sites. Is that understood? So you see several restriction enzymes, and here on this plasmid it is indicating the recognition sites. All right, where these enzymes would recognize and eventually cleave. Couple things to note here, and we will discuss this in a while. This here is ORI. ORI stands for origin of replication. So remember that in prokaryotes, all these genetic material, which are circular, have one origin of replication, where when it's time for replication, there is where it will start to create that replication bubble, and replication would proceed bidirectionally. Okay? So all these plasmids must have that origin of replication. There are other things that we will discuss in just a few that you'd find on this plasmid as well. Here we have AMP, that stands for ampicillin, and that is actually a selectable marker gene, all right, whereby you'd be able to select transformants whether those genes are being expressed or not. Plasmids have these following names. So whenever you're reading literature and you see a small b followed by a couple capital letters and a number, that's the name of a plasmid. This, what we're seeing here, is a plasmid, but it is also referred to as a construct, right? Because these plasmids can be actually designed by people. They can decide what to put on that particular plasmid. So they're also referred to as a construct, because these are constructed, right? So, and what you see here is the size of this plasmid, 4,363 bases. We're talking about nitrogenous bases in that circle, right? Here we have another example of another plasmid. Watch the name of this one, small p, capital UC, and the number. All right, that's an indication that that is the name of a plasmid. And this one has a size of 2,686 base pairs. And here it has the indication of all the restriction sites that are on this plasmid. These must have three features. One, an ORI sequence which directs replication. I indicated that in the first diagram. So the ORI sequence must be there because there is where the replication bubble will form when it's time for replication. Okay? A dominant selectable marker, these are usually genes that confer resistance to antibiotics. I showed you guys the ampicillin gene. All right, that was in purple. That is the ampicillin gene. That gene confers resistance to ampicillin, and that is used as a selectable marker gene. It simply means that whenever you're doing any genetic transformation, the gene of interest will always be there in that plasmid as well. And so when you culture these cells that have this transformed plasmid in it, you would add ampicillin to your culture media, for example. And if your cell is actually transformed, meaning that it has that ampicillin gene, then it would survive that culture media that has the ampicillin in it. If it is not transformed, then the cell will eventually die. So it is used as a method of selection. Right? So the selectable marker gene aids in selecting those cells that have been transformed from those that haven't. All right, so that's the purpose of the selectable marker gene. Third, unique restriction enzyme cleavage sites. All right, remember these are the sites on that plasmid where these restriction enzymes will recognize and cleave. So unique restriction enzyme cleavage sites for the insertion of the DNA sequences that are to be cloned. Right? So it's important that you have these restriction cleavage sites so that these restriction enzymes recognize where they are, cleave there, insert that portion of DNA that you want to clone, and then with DNA ligase, that portion is spliced. Okay. Generally, plasmids have names that begin with a small p, all right, like what we see here. So small p, couple capital letters, and a number that follows. All right, so whenever you're reading in literature and you come up to these, you know that you're talking about a construct or a plasmid, all right, that has been designed by a genetic engineer. Of course, you can put whatever gene of interest. For example, if your objective is to produce insulin in the safflower plant, then a gene for insulin would be placed. That would be placed in the plasmid, right? If the objective is to produce a plant, a banana plant that is resistant to a fungal disease, then that gene that confers resistance to that particular disease would be placed somewhere there in your plasmid, along with the selectable marker gene and so on. Okay? Second cloning vector is bacteriophage lambda cloning vector. When you see this term, what comes to mind? What is this? What is a bacteriophage? It is a, fill in the blank, one word. Thanks, Johara. It is a virus, right? It is a virus. And sometimes they're referred to simply as phage. So bacteriophage lambda cloning vector, all right? So because of viruses' natural ability to infect other organisms, then they can be used as cloning vectors in order to clone certain segments of DNA, all right? So the lambda cloning vectors possess restriction enzyme sites so that DNA fragments can be cloned using them. One example is the lambda replacement vector, where there is a left arm and a right arm that collectively contain all the essential genes for the lytic cycle. Between the two arms is a disposable segment of DNA. It doesn't contain any genes needed for phage propagation. And so this disposable segment can be excised, cut out, and be replaced with a DNA molecule from a different organism. All right, so if we look at this diagram here, we will understand how you can use bacteriophage lambda as a cloning vector. Okay. So this right here is the right arm and the left arm. What you see here in green at the top would be non-essential region. So with restriction enzymes, it would cleave this molecule right here. All right. So that non-essential region would be cleaved and do away with. And then the foreign DNA that you see here in red or pink or whatever color that is the portion that will be replaced. So you'll replace the non-essential region with your foreign DNA. That foreign DNA comes from a different organism. All right? So you end up with this recombinant DNA molecule that has the right arm and the left arm from the bacteriophage, but it also has that foreign DNA. All right? So there you have what is called the hybrid DNA or recombinant DNA molecule, combining genetic material from two different organisms. So that hybrid DNA that we see here is then packaged inside the phage. So what you have here now is an infective phage particle. So that when that phage go and infect that bacterial cell, it will actually transfer that foreign DNA into that bacterial cell. And then that bacterial cell can now, when it replicates, it is also replicating the foreign DNA. What actually happens here, guys, is that the phage injects this foreign DNA into the bacterial cell, and it hijacks the reproductive machinery of that bacterial cell. So when that bacterial cell is reproducing, it is also making more copies of that foreign DNA that you actually wanted to clone. All right? So this is the bacteriophage lambda cloning vector. So what we have here in the next slide would be explaining this process of what takes place right there. Okay? So it says first, the lambda vector is cut or cleaved with the restriction EcoR1, the restriction enzyme, right? EcoR1 to separate the two arms from the replaceable segment. That replaceable segment was the segment in green, that is a non-essential region. DNA fragments to be cloned are generated. That was the portion in pink. This foreign DNA fragment are then mixed with the lambda DNA fragments, and the pieces ligate together by DNA ligase. Only about 15 kilobases of DNA can be inserted into a lambda replacement vector. So lambda cloning vectors are used for initial cloning of genes of interest. Okay? The third cloning vector is the cosmid cloning vectors. Plasmid and lambda cloning vectors can only be used to clone small DNA fragments. So we're talking between 5 to 10 kilobases in plasmids and about 15 kb in lambda. Some genes may be as long as 30 kbs, hence cannot be cloned in one piece using these cloning vectors, the plasmid and the lambda. And so when you have a gene that is much larger, let's say 30 kbs, then you have to use a different cloning vectors, such as the cosmid cloning vectors. DNA fragments of about 35 to 40 kbs can be cloned in vectors called cosmids. Unlike the first two, cosmids do not occur naturally. They have been constructed by combining features of both plasmids and lambda. All right, so plasmids and lambda, those are naturally occurring cloning vectors. However, cosmids are cloning vectors that are actually put together by genetic engineers. All right, so they're able to build this cloning vector. A cosmid has an ORI sequence, that is the origin of replication, right? Where replication will start. It also have a selectable marker, and of course, unique restriction sites, similar to the first one, the plasmids, right? So it has all these three components: the ORI sequence, a selectable marker, and unique restriction sites. Apart from these, they have COS sites, which permit packaging of DNA into a lambda phage particle that facilitates the introduction of large DNA molecules into bacterial cells. All right. So you have these cosmids that will be prepared, and of course, the lambda phage will aid in transferring these cosmid vectors into bacterial cells. So the basic features of a cosmid is what you see here: origin of replication. It has restriction enzyme sites. It has the COS sites, and it has the selectable marker gene, the antibiotic gene. All right, so that is the selectable marker gene right there. All right, so here we have a cosmid. What will happen is that you'll have cleavage at this polylinker right here. So restriction enzymes would cleave right here at the polylinker. So it will convert this circular molecule into a linear molecule. So after cleavage at the polylinker site, all right, so here we have the origin of replication, you have the COS, and here you have the selectable marker gene, which is ampicillin in this case. All right, the gene that confers resistance to ampicillin. So after cleavage, it converts this circular DNA molecule to a linear molecule, and you end up with this right here on the right. So a linear molecule having the COS site there as well. These cleaved cosmids, which are now in linear form, are combined with the target DNA molecule that you want to cleave. And so you end up with, these two are mixed, right? And so you end up with a recombinant DNA molecule comprising of target DNA as well as the cleaved cosmid. So this recombinant DNA molecule is packaged inside that phage, and that phage is capable of infecting bacterial cells, all right, by inserting this recombinant DNA molecule inside the bacterial cell. And of course, this will eventually use the bacterial cell's reproductive machinery to make more copies of itself. Okay? So this is how the COS vector operates. Remember that the advantage here is that this has the ability to clone segments of DNA that are much larger, 35 to 40 kbs, than what plasmid and lambda, bacteriophage lambda, can actually do. The last cloning vector that we look at is the shuttle vectors. The first three vectors must be used to clone DNA within E. coli cells. Other vectors have been developed to transform mammalian cells, other animal cells, plant cells, and yeast cells. A shuttle vector is a cloning vector that allows it to replicate in two or more host organisms. So here we have an example of a shuttle vector specifically for yeast. And here we have, all right, all these must have these things: origin of replication. And here we have the selectable marker gene. And of course, you will also have a restriction sites on this vector as well. All right. So these shuttle vectors are used mostly for cloning mammalian cells, plant cells, yeast cells, and so on. All right. So that is the difference between this and the first three. Okay. All right. We're talking about cloning, making more copies of a segment of DNA, and we can use bacterial cells in order to clone portions of DNA. But what you see here, polymerase chain reaction, or PCR, this is a biotechnological technique that can also be used to make more copies of a DNA segment. So this is what this is used for. All right. Of course, this is used as a diagnostic tool as well. I'm sure during this pandemic you've heard of PCR tests and rapid tests. All right. That same PCR test that they were talking about, that is more reliable, is what we're going to discuss. And so we'll understand the concept of PCR and how it is used to diagnose certain diseases. All right. But the overall concept of PCR is that it mimics DNA replication. So whenever DNA replication is taking place naturally in cells, you can actually make more copies or replicate DNA segments using PCR in the lab. Okay. PCR is a method for selectively and repeatedly replicating defined DNA sequences of a DNA mixture. Okay. Mixture contains the DNA sequence to be amplified and a pair of oligonucleotide primers that flank that DNA sequence. I hope you can recall our discussion about DNA replication. All right, in DNA replication, what was the next step that took place after denaturation? So after denaturation, when that molecule opens up into single strand, what takes place after that? Can anybody tell me?
Dr. Daniels, isn't that when the, I think, RNA polymerase binds to the molecule to start the replication?
It wouldn't be the RNA polymerase, but it will be the RNA primer. So you have some short pieces of oligonucleotides, which are called the primers, those anneal to the two strands of the DNA. And it is from that point where that RNA primer is that the RNA polymerase will start to extend that complementary strand. And remember, we said that the leading strand goes in the direction of the replication fork, and that occurs continuously, while the other strand must be done in fragments called Okazaki fragments, because it has to wait for denaturation of that molecule, all right, and then the primer to anneals before it can build in the opposite direction of that replication fork. It is important that you understand DNA replication that occurs naturally that we discuss for you to understand how PCR works, because I said that PCR mimics DNA replication. Okay, so it says the mixture contains the DNA sequence to be amplified. When we use the word amplify, we mean make more copies of, and a pair of oligonucleotide primers that flank the DNA sequence. All right, so you need RNA primers in DNA replication, and of course you need primers in PCR as well. When you are going to use PCR as a diagnostic tool to diagnose, for example, COVID-19, you must use specific primers that will be able to anneal at specific genes on that RNA molecule of the virus. So you have to use specific primers that will recognize genes on that viral molecule. So as long as the virus is present in this PCR reaction, those specific primers will anneal to those spots, and it will make more copies of that DNA segment. So when you see more copies are being made, and this is done in real time, it can be done in real time, you can actually see it on a screen, then that is an indication that the virus is present. If the virus is not present, those oligonucleotide primers will find absolutely nowhere for it to anneal, and so PCR will not take place. It won't be able to make more copies of that segment because the primers cannot find the complementary strand where it can anneal to. Is that understood? Do you guys understand how you can use this to diagnose? So you need specific primers to diagnose specific diseases. Okay? So these are the steps when doing PCR. And I want you to reflect or have it in your mind, in your background, the steps that occur during DNA replication. So remember, the first thing was the strand opens up, right? Denaturation. Then you have annealing of the RNA primers, and then you have polymerization taking place, whereby the DNA polymerase extends the complementary strands, all right, to form that double-stranded DNA molecule. So the first step in PCR is denaturing the DNA to single strands, and that is done by incubating at 94 degrees Celsius. When DNA replication takes place naturally, what does this job? Denaturation. Tell me, what does the job of denaturation? No, it's not heating. We're talking about natural DNA replication that takes place in your cells. What does the job of DNA naturation? What helps to open up that DNA molecule and convert it to single strands? No, it's not the RNA primer. Hydrogen bonds, no. Hydrogen bonds are the ones that have been broken. My question is, what helps to break these hydrogen bonds to convert that and denature that molecule? It is not the DNA polymerase, no. A test coming up Thursday. Test is coming up Thursday. And this material will be evaluated, not hybridization. It is an enzyme that helps to denature that DNA molecule. Thank you, Marva. DNA helicase. Helicase is that enzyme that helps to denature the DNA molecule. That is what takes place when natural DNA replication occurs, right? In PCR, we do not use helicase. Hydrogen bonds that hold the two strands together are weak and they can be broken with heat. And so in PCR, that DNA double-stranded is converted to single-stranded by incubating the reaction at 94 degrees Celsius. So heat is used to do the job of the helicase. Remember, this is PCR, this is done in a lab. But it mimics what takes place in nature when it comes to DNA replication. All right? After the DNA molecule is denatured, so we have single strands now, then you have annealing of the specific pair of primers that flank the targeted DNA sequence. So you remember you have to have specific primers that will anneal to the specific area that you want to amplify. Okay, so you have to have specific primers. After the primers are annealed to the two strands of the DNA molecule, then you have extension. So they extend the primers by DNA polymerase. So DNA polymerase helps to extend that from where the primer is annealed and builds the complementary strand of those two molecules, those two strands. Now, we're talking about DNA polymerase. This is an enzyme that will be responsible to catalyze the polymerization. But we heated this reaction at 94 degrees Celsius. Folks, what happens to proteins and enzymes at high temperatures? They become denatured. So because of the fact that we're using high temperatures in this reaction, the normal DNA polymerase cannot be used because this would become denatured. It won't be able to do its job. So a special DNA polymerase, which is called Taq polymerase, is used because Taq polymerase can withstand extremely high temperatures. And this enzyme, Taq polymerase, is isolated from the bacterium called Thermus aquaticus. So that enzyme is isolated from this bacterium called Thermus aquaticus. And the one characteristic of Taq polymerase is that it can withstand high temperatures. So it would still be able to do its job in polymerization, extending the complementary strands in this reaction. So these are the three major steps of PCR. And of course, these steps are repeated. So from A to C would complete one cycle. All right, one cycle. So when you run it over again, that's two cycles and so on. So these are repeated until all the components of the reactions are exhausted. We're talking about the DNTPs, all right? Until all of that is exhausted, then the reaction stops. one molecule is produced exponentially. So if you have one molecule, at the end of one cycle, you end up with 2. And at the end of the second cycle, you end up with 4, and so on. All right? So it is produced exponentially. The equipment used for PCR is called a thermal cycler, and the procedure is susceptible to contamination. So it has to be done in a very clean environment. You have to use gloves. All your pipette tips that you use, you have to dispose of them as soon as you're finished. All right, so it is susceptible to contamination. This diagram here outlines what takes place during a PCR reaction. So what you see here in green that is the portion of DNA that you want to amplify or make more copies of. Of course, you will need the 2 primers that will anneal the 2 strands. Besides that, you will need the DNTPs, the nucleotides, all right, to build the complementary strands. Excuse me. So the first step that takes place is what? Denaturation. All right? Opening up of that double-stranded DNA molecule. And that is done at high temperatures. We're talking 94 to 96 degrees Celsius. The second step is for your primers to anneal what you see right there. Your primers to anneal to the exposed bases in these 2 single-stranded molecules. So that is the second step, annealing, and that is done at a lower temperature, around 68 degrees Celsius. Remember I said using PCR as a diagnostic tool, these primers that you see there in pink, those would be specific primers that can actually, that have the complementary bases for specific disease. For example, if we're diagnosing COVID-19, these primers would have the complementary bases that would recognize certain genes of that virus, right? So if the virus is present, these primers would find the complementary bases and anneal there, and PCR will proceed. And then when you see that large number of molecules being produced, that is an indication that the test is positive. If these primers are mixed with that DNA and it cannot find its complementary where it can anneal, PCR reaction will not take place, and then that test will end up being negative. Okay, so here we have primers being annealed, and once it anneals, then it moves on to the third step, which is elongation or extension, and that is done at approximately 72 degrees Celsius, whereby the top polymerase will be the one to build the complementary strand, this one in this direction and this one in this other direction. Remember that these are built in the 5' to 3' direction, right? So here, this would complete one cycle. And so here it would go again. And so it would complete another cycle here. And you can have endless cycles taking place based on how much DNTPs you have in your reaction. Okay? So components for PCR, you have to have your DNA sample. This is the sample that you want to amplify, make more copies of. You need your primers, the specific primers of that DNA segment you want to amplify. You need your top polymerase. That's the enzyme that will facilitate the polymerization. These are your nucleotides, your DNTPs. These are required for the top polymerase to help build the complementary strand. All right. Of course, all of these will be put inside this mixed buffer, and all that reaction will be placed into this PCR tube. This PCR tube will then be placed inside this thermal cycler, whereby here you will set your temperature for denaturation, your temperature for annealing, and your temperature for elongation. So all those parameters will be set on your thermal cycler. Once all those are set and your PCR tube with everything is inserted in the machine, press start, and that's it. In the case of real-time PCR, this machine, this thermal cycler, is connected to a monitor whereby you'll see a graph. And when you see that graph is going up, it's an indication that more copies are being made, which would indicate a positive test. All right? So here we have the process. This is in one cycle. So you have denaturation of this double-stranded DNA molecule. This is done at high temperatures, right? So once these strands are separated, then you have annealing of the primers. So you have one primer here and one primer here. So once those are annealed, then you have the extension. All right, so extension, whereby it will build upon starting from the point where the primer is and build the complementary strand until you end up with 2 new copies of DNA molecule. All right, it's 9:14. My time is up. Next time we meet, we will just review PCR, and then I'll show you a video on PCR. Any questions before we leave?
So meaning that we're not meet Thursday for class, right? Or we still
Oh, yes, indeed. Thursday we'll meet, yeah, because the test is 4 o'clock. So yes, we're having class on Thursday.
No, sir, but we could use that time to study.
Yeah, we could use tonight for study too.
No, sir, I have one next test for study still. I have one next test for study first. Sir. Thanks, sir. Bye, sir.
All right, folks. Have a great day.